I have one announcement to make before the next speaker. So there is a box here which has been put there. If you have any open questions about psychedelics or regarding this conference, you can put your questions there. Someone is going to answer that this afternoon at 4 p.m. in the audience. In the auditorium. So please feel free to put your questions there. This is the announcement that I wanted to make. With this, I want to welcome Dr. Kate. Can we have some silence, please? Yeah, thank you very much. I would like to invite Dr. Kate Godfrey, who is a postdoc in the UK. She is one of the 3 selected talks, and I'm really, really happy and looking forward. Most of the talks here are from invited speakers. Kate is one of the selected talks, so I'm really looking forward to the high quality research that you applied with. Her title would be Using EEG to Measure Human Neuroplasticity, which is a big topic if there is a correlate of neuroplasticity in psychedelic research. So welcome, Kate.
Hello? All right, well, I'm in a terrible position of having to follow that talk, so I apologize. I'm honored to be here. I'm honored to be in the slot. This is an amazing opportunity. What I'm going to be presenting about is very similar to what Franz just presented about, arguably at a more basic level, so bear with me a little bit. But I'm going to be talking about EEG and how we can use EEG in psychedelic research to look at neuroplasticity and also predictive coding. So very similar to what was just presented. So I work at Imperial College. We do a whole bunch of different studies in healthy people and also clinical populations. And my talk today is going to be a little bit about how we do integrate these sort of EEG paradigms into our clinical studies to look at, because these sort of measures are not only implicated in psychedelic research, but also just general psychiatry and psychiatric conditions, and how we can potentially use them to index recovery as well as the effects of the psychedelic drug itself. So, very brief, crude introduction to the predictive processing model of brain function. So as we go about our lives, we're constantly integrating incoming sensory information without sort of prior beliefs or sort of top-down processing. It's a very efficient way of sort of processing a lot of sensory information. If we were to sort of attribute everything with the same sort of salience, we would very quickly be quite overwhelmed. So this way, only the sort of relevant, salient, sort of surprise stimuli sort of taken up the hierarchy and integrated into that model with the top-down sort of inferring of what we sort of expect to be around us. And that's where that sort of concept of perception is controlled hallucination. We're sort of generating what we sort of think is around us. We have the error detection, which goes up the hierarchy, and that's what I said, like the surprise response. And then we have the model updating going down the hierarchy. So that's from the priors. They take in that new information, they update, sort of like, okay, well, I need to update my model, and they infer that down onto the model, and then we have our model that's sort of eventually calibrated, and we, over our lives, build these sort of models at different levels of the hierarchy as well. So from a very low level at the sensory cortices to all the way up to the sort of higher level, like system-wide models. In psychiatric conditions, this is all sort of quite theoretical, and I'm sort of going to talk about how we can maybe use these tools to provide a bit of evidence for them. I'm aware that the evidence currently isn't like super robust. But we could think of psychiatric conditions as having sort of a heavily weighted prior and incoming sensory information being sort of not as willingly updated into the model. So if we have a very strong prior belief, such as I am unworthy, and then we have contradicting sensory information, such as positive reassurance, that's feedback. It's not integrated into the model in the same way that it would sort of be in a healthy brain. And those prior beliefs more persistent, less likely to be sort of updated, and we have sort of a more rigid model that is harder to break out of. Then when we talk about psychedelics and predictive processing, particularly with the Rebus model, we then have sort of the opposite, where there's a relaxation of prior beliefs. They are producing sort of less of an influence onto the model, and the incoming information has a higher weighting. And so this potentially could underlie how these drugs have these sort of therapeutic benefits by updating this sort of balance of top-down and bottom-up processing and allowing this sort of contradicting information to enter into the space to update the model, redefine those prior beliefs, and produce sort of therapeutic benefits. And that's what we see here, sort of that readjustment of those sort of two pathways. And that model calibration, or that sort of persistence of this effect, that's where the neuroplasticity really comes into, is that updating of the model and the reinforcement of these new sort of thought patterns, and the evidence for psychedelic-induced plasticity in the rodent model is becoming more and more robust. Did some excellent talks on that here, and yeah, I'll sort of talk about that here. So in vitro and in vivo, I've really enjoyed following the preclinical literature around psychedelic-induced neuroplasticity. And then when we talk about a therapeutic response, we also see these long-lasting changes and these drugs having like lasting effects. But there's this missing step in the middle, where currently the evidence in humans for enhanced neuroplasticity with psychedelics is limited, and our tools to investigate this are also sort of limited. What we do have available to us to measure human neuroplasticity, PET is a really good example. So, for example, we're using at Imperial the UCBJ marker of synaptogenesis. You can check out Gabriella's poster and the lunch break. That's in a ketamine study. So that's very exciting and a potential marker of this increased plasticity. There's also structural MRI and DTI. There was a great talk about changes in DTI yesterday, and then also we've got exciting upcoming results from our INSIGHT study. We're seeing lasting changes in DTI in healthy individuals following psychedelics, but I won't really talk about that here. There's BDNF, which I think when measured peripherally has very mixed results, and the validity of it as a marker of like the CNS BDNF is sort of questionable, but it is available as a potential marker of neuroplasticity. And then we have EEG, which is where I come in. So I think EEG is it's a really interesting measure. It's quite often overlooked. It has the sort of sexy older brother MRI, and EEG is kind of forgotten about. But there's really interesting ways that we can use EEG to look at quite quick changes in brain function at quite a sort of granular level. Very broadly, an ERP, this is what an ERP is. I'm aware that not everybody knows what an ERP is. So you take the EEG, you put it on, you mark when a sort of sensory presentation occurs, and you average that signal. Then you get to average out all the other noise, and all you're left with is the brain's response to that sensory information, whether that be a visual stimuli or an auditory stimuli or a tactile stimuli. This is what the sort of brain's evoked response is, and then we can look at the magnitude of that evoked response, and we can try and manipulate the magnitude of that evoked response. In psychedelics, they do tend to, just by themselves, modulate sensory perception. So acutely, under the influence of a psychedelic, generally ERPs, I think, are suppressed depending on the sensory information. But what I'm kind of going to be talking about is more using these post-acutely. So in the hours or day following the drug administration, you don't have that same sort of confound of the sensory perception being altered anyway. They're back to a normal state. And then we can try and like manipulate these ERPs and use it as an index of brain function. The first task that I'm going to talk about is the visual long-term potentiation task. There's sort of iterations of this task around, but this specific one was developed at the University of Auckland, which is where I'm originally from. And it involves presenting visual stimuli. So you do different blocks. You have a baseline block or a pre-tetanus block, and that's just a 1 Hertz stimulation. So you put a circular sine grating on the screen one every second for about 4 minutes. And you're just getting the brain's sort of baseline responsiveness to that visual stimuli. We then introduce a tetanus phase. And so that tetanus is sort of derived from the animal literature where you do like tetanus stimulation electrically of either a cell culture or in a mouse or rat brain. But we do it visually, so it's a 9 hertz, so 9 per second fast flashing presentation of this. And that fast flashing is thought to elicit this LTP process. So that's the cells that fire together, wire together, by the Hebbian sort of plasticity model of increasing your plasticity or sort of enacting that neuroplastic change, and meaning that those units that are being activated will be more readily activated the next time they are initiated. So we do this tetanus block, it's 2 minutes, it's quite interesting, just a fast flashing. And then have a little break, and then you do the baseline again, basically, so the 1 hertz stimulation. And we can see how the ERP is potentiated. So this is an example here. You've got the pre-tetanus in the blue, and then the post-tetanus in the red. And you can see that ERP, the evoked response, is increasing, so indicating potentially more units are being activated, or they're sort of more readily firing, and you're having a larger recruitment of cells and a stronger response following that tetanization. We have evidence of this in sort of a clinical sense with ketamine. So there was a study done a couple of years ago at the University of Auckland using ketamine and remifentanil as active control as a treatment for major depressive disorder. It was a crossover design, so it's within subjects. And what was found is that ketamine did increase the plasticity, or the sort of recruitment, or the degree of potentiation, I should say, following, it was 3 to 4 hours post the ketamine infusions. They were complete, they were sort of back to regular sort of sensory perception levels. But yes, ketamine caused this enhancement in the degree of potentiation that was elicited, and indicating that LTP was being more readily activated by the tetanus stimulation. This hasn't yet been done with this paradigm in psychedelics population, but we're currently using it in a lot of our studies. So you can see here we're doing it in our continuous infusion work, 3 to 4 hours post DMT continuous infusion. We're doing it in our clinical studies, looking at psilocybin for anorexia, fibromyalgia, and OCD. There's also a few studies coming up using it in microdosing. So there's one at the University of Auckland, which was 2 hours, sort of like in that acute phase. And then we're also running one at the moment where we're going to do it a little bit later as well, to see sort of the comparison there, as when's the sort of best time to do this. But yeah, these are all sort of going to be coming out in the next year or so it's very exciting. The second task I'm going to look at is the roving mismatch negativity. We've already heard a little bit about mismatch negativity, so I won't go into it too much. But yeah, you present a series of tones, and you're changing the frequency or the pitch of those tones. So you become sort of habituated, right? If you have a series of tones, you're going to sort of, the first one might be novel, and then they increasingly become less novel. You change the pitch, and suddenly, oh, that was a surprise. And so we have these deviant tones and the standard tones. The roving paradigm, I should say, is kind of unique from other mismatch negativity tasks, because sometimes it's a continuous, there's sort of a standard frequency and then a deviant frequency. So you have the standard tone, and then you have a deviant, and then you go back to the standard tone. The roving offers a really unique opportunity to look at that model updating, because the standard tone is actually the same pitch as the deviant. So it's not that we're returning to a standard tone, it's that the model is being updated to say, okay, this frequency is now usual, and we can go back to sort of not attending to it. So in that way, we get to look at not only the mismatch negativity response, but we also look at the repetition suppression. So how quickly is that model updated to say, okay, this is no longer a surprise, this is now a standard tone. And you can see that here, we've got the mismatch negativity, where you can see in the dark blue, there's a big sort of difference in the ERP, where there's that surprise elicits a greater response. And then we can see the repetition suppression. So the mismatch negativity, as you can see, is negative in that first tone. It has its biggest effect, and then it increases back to usual sort of standard response. This has been done acutely in LSD with our group. So Chris Timmerman published this several years ago with Meg Spriggs. It was 20 participants, placebo LSD, it was a MEG study actually, which is similar to EEG, but using the magnetic field rather than the electric field. And what was found was that the MMN was decreased, but the standard was sort of, if anything, stayed the same or was slightly increased. So even though this was acute, the standard wasn't sort of being impacted. Potentially this is an auditory thing rather than a visual thing, or yeah, it depends. But the mismatch negativity was reduced, and that was sort of interpreted as that reduction of the model's sort of top-down influence. So it's less of a surprise. If the model isn't exerting like, this is what the next tone should be as strongly, you're going to be less surprised when the tone differs from the one you sort of expect. In the psilocybin literature, it's a little bit different. So generally in psilocybin, the MMN hasn't really produced, like the MMN alone, hasn't produced robust changes. There's sort of conflicting evidence there, which is quite interesting in itself. Potentially the difference in the sort of receptor binding between these drugs may underlie a slight difference here. But I would be really interested to look at the repetition suppression with psilocybin. See, potentially what if the MMN may not be changing, but the model updating might be, and that's an interesting question. We also have evidence in this in a clinical study. So this is the same one, the same ketamine study that was done at the University of Auckland. And here the MMN was increased post-acutely after ketamine. Which is sort of the opposite direction to that acute. So it's saying, all right, well, acutely, the MMN is suppressed, but post-acutely, there's potentially this rebound effect where the MMN is increased, and that could be that sort of transition, like I mentioned, with the psychedelics sort of balancing out that balance of top-down and bottom-up processing, and sort of having a bit of a rebound effect. And we're hoping to sort of look into this with the psilocybin studies as well. Yes, again, the study is currently ongoing. So together, overall, I present these two sort of paradigms as an interesting sort of battery to probe neuroplasticity and predictive processing in the context of psychedelics and also in the context of clinical conditions. So by using the MMN, we can look at error detection and model updating. That's using the MMN response and the repetition suppression. And then we can also look at the sort of long-term calibration or the neuroplasticity in a more sort of general sense using the visual long-term potentiation task as a proxy for the sort of plastic state of the brain. The visual cortex is obviously not an ideal candidate in terms of the action of the therapeutic effects of psychedelics, but it is a cortex that we can quite easily probe. So we use it as a proxy measure. And then this maps onto potentially the trajectory of the response to these treatments. So if acutely, there's sort of that error detection being affected, short term, there's an increase in sort of the ability for the models to be updated, and then long-term calibration occurs a little bit later with that increase in plasticity, and that's sort of underlying the persistent changes. I'd like to thank everybody on the slide. So this is the Center for Psychedelic Research, as well as my supervisor, Suresh from Auckland, and Rachel Sumner, who did a lot of the ketamine work. And yeah, thank you for your attention.
Yeah. So any questions from the audience or the online audience? Yeah? I pass you.
Hi, thank you. You mentioned it briefly, and I understand the limitations of looking at neuroplasticity in humans, but can you say a little bit more as to what you think, to what extent you think that we can draw any conclusions on neuroplasticity based on primary visual cortex neuroplasticity?
It definitely depends on, like, plasticity is such a broad word that I think encompasses like a lot of different processes, and it means different things to different people depending on what they're researching. I think the visual long-term potentiation task offers like a small slither of the pie of plasticity. It's interesting because these drugs obviously have like really broad pharmacological action across the cortex, and they do have a lot of action in the visual cortex. So if it is a really like mechanistic sort of like circuit level change in the ability to elicit LTP that is occurring in these drugs, then yes, we could detect something that could indicate that sort of like really biological sort of eliciting of an enhancement of LTP. I forgot to mention also, because ketamine is sort of potentially a better candidate for this because of the primary action on the NMDA receptor, which is the sort of primary mechanism behind LTP. So it will also be interesting to see if the downstream sort of glutamatergic action of psychedelics can enact the same sort of effects. So it's sort of more that sort of sense of the really sort of mechanistic biological things. But it definitely, I think, in absence in LTP task, potentiation difference doesn't necessarily equate to an absence of any plasticity enhancement of psychedelics. It's just one sort of bit of it that might help uncover like the scope of the psychedelics and their effect on plasticity.
Okay, thanks.
Yeah, thank you for your talk, very interesting. I have a bit of a follow-up question, I think, to this, because I found in the first talk today very interesting how Professor Dühlen talked about more of a social learning that's increased. And I was wondering if you know of any paradigms that measure LTP, neuroplasticity, in more of a social way, like, yeah,
Not necessarily. I know there are like definitely like EEG tasks that can be done to probe like other aspects of cognition, including like social behavior. It's less so because MRI is sort of more suited for something like that, I think. Because EEG, this is the strength of EEG, being really like quite granular, like sensory processing level, provides really good like quick and quite robust signal around those sort of fast changes. When it's something that's sort of more integrated across the whole brain, it's a little bit harder to then look at that with the EEG. So potentially that would be more in the realm of MRI research. Yeah, but no, I agree. That was a really interesting talk. Yeah, it's fascinating stuff. Yeah. It's okay.
Any other questions? Okay. Can you pass the mic there, please?
Oh, sorry, I can't.
You talked about long-term potentiation, but have you looked at through prism of LTD, long-term depression?
Yes, so that is an interesting, the two go hand in hand. So the question, if anyone didn't hear, was about LTD as well as LTP. So they're sort of the two ends of the spectrum of this plasticity. Arguably like the 1 Hertz potentiation, like 1 Hertz stimulation is used to elicit LTD. And it is something that's sort of been looked at. The duration in which we do it is typically not sufficient to elicit any LTD, but it is very feasible that these drugs could not only enhance LTP, but also LTD. So yeah, while this paradigm is set up to measure LTP, it would be interesting, and it could be used in the reverse to look at LTD, if you potentially did a longer stimulation block of a 1 Hertz suppression. Cool.
Oh, the clapping. I think there was another question. Can we take that question there? Yeah.
Yeah, thanks. I was wondering if you've ever either studied or thought of studying in the mismatch negativity. You mentioned compounds that alter the perception of pitch, like carbamazepine or diisopropyltryptamine.
I haven't done it. I haven't necessarily considered it a whole lot either, but it would be really interesting. And that's why I think that, like what I mentioned with the acute effects, potentially there's a sensory, when you've got a sensory perturbation as well as the task that you're looking at, it arguably would sort of complicate things. But it could be interesting to look at to see if the like discrimination between pitches or the sort of surprise responses within a normal tone would be. But I think it would probably require a more tailored paradigm for that rather than this paradigm. It would need to be considered, how to implement that.
Thank you.
So thanks, Kate. Thanks so much for the talk and the question answers.